Welcome, everybody. The Office of Programs to Enhance Neuroscience Workforce Diversity within the National Institute of Neurological Disorders and Stroke is pleased to welcome you to the Open Stage monthly webinar series. This is a targeted outreach effort for our diverse neuroscience community to learn about NINDS programs and network with NIH and NINDS program staff. Each webinar will feature a different NINDS program, division, and cluster, all with the goal of increasing the representation of diverse groups in our portfolio. So this month, we are very excited to hear about drug discovery and development, funding and training opportunities from NINDS program directors in the Division of Translational Research, as well as NINDS awardees. So before we get started, I just have a few housekeeping items. If I could go to the next slide, perfect. So a few meeting reminders. All attendees are initially muted. Please submit questions using the chat function, and a reminder that this meeting is being recorded and will be transcribed and archived on our NINDS website. You can enable closed captioning under the transcript at the bottom of your screen. And details about where to find this archived version will be posted on social media. We will send you all emails after the webinar is over with all the resources and everything, slides and everything. So look out for that, and please do stay connected with us and feel free to email us if you have any questions about this webinar series. So with that, I will pass it off to Dr. Carolyn Bonder to get us started.
Hello, welcome. First, we're going to have two speakers today. One is Program Director and Director of Small Molecule Neurotherapeutics Development, Dr. Charles Sywin, and we'll also have Program Director Miriam Pellimonter for the second half of the talk. So Chuck, you want to get started?
Sure. Next slide. So the National Institute of Neurological Disorders and Stroke, also NINDS, seeks to have fundamental knowledge of the brain and the nervous system, and to use that knowledge to reduce the burden of neurological diseases for all people. And what really is highlighted I've been working for NINDS for a little over a decade, is, the burden of premature death, disability, and suffering from neurological diseases is probably greater than any other group of diseases out there. And, and certainly there is many challenges. They are real, and they cry for solutions. And I can tell you from my personal life, like many others, since joining NINDS, I've lost many extended family members to neurological diseases in the last decade. And so it's come home for me that, what I do every day impacts the people around me. Disclaimer, any opinions you hear today are my own or those of the presenters, and not necessarily those of the government. I have no conflicts of interest to report for myself or for Marianne or any other speakers today. The goal of today's presentation is to provide a brief overview of the NINDS organization, provide an overview of translational research in general, highlight a couple of key programs that NINDS hosts that are trans-NIH in nature, the Blueprint Neurotherapeutics Network for small molecules, as well as the HEAL Pain Therapeutic Development Program. We're going to try to hopefully increase your awareness of funding for these kinds of neurotherapeutic development activities, as well as for training opportunities to move your discoveries towards the clinic. And at the end, provide you some helpful hints for successful applications to our translational programs. So there are 27 different institutes and centers at NIH. 24 of them award grants. And what you really need to know about them is each one has different missions. It has different funding priorities. It has different budgets, different types of grants they support, different procedures, different funding strategies. So despite the fact we're NIH, you have to really understand what the institutions are within that have the disease area and the mission that you need to go under for your neurotherapeutic development activities. So talking to NIH from the onset is a great idea, and we can help get you to where you need to go and to find the right people to talk to and to educate you about the opportunities. And this is one of those things we're doing today. Next slide. The application process is many steps. It starts with you. It always starts with you coming in with a research idea, and then doing the process by which you can apply for an application. You need to be properly registered and have your eligibility confirmed. You need to submit the electronic version of your grant. We don't take paper anymore, it's all electronic. So you got to be in the system, all registered to go, file electronically. It then goes to the Center for Scientific Review, who assigns it to an IC. So that discussion I had in the last slide about 24 ICs funding, part of that is you can sort of help direct it to where it should go by helping review and giving sort of your selection of where you think it should be assigned. It'll be assigned to a review group, either at the CSR or at IC, an SRB at individual center or institute. The scientific review group, back one slide, continues to evaluate the scientific merit, and then it moves on through advisory committee approval. IC staff prepares a funding plan. The IC director determines the funding. We allocate funds, and we finally award the grant for you to conduct your research. So this process can be lengthy in this, and some are faster than others, different mechanisms. So again, talk to us about what your needs are, and we can tell you a bit about what the time constraints are that we're under. Next slide. So start planning early. And this is a great little tool set here. NIH Reporter could help you with matchmaking and help you build your team. NIH Reporter could help you find your program director. You can utilize the Find Program Director NINDS website as well as our funding opportunities. But NIH Reporter is a wonderful tool. It can help you find similar projects. It can help you find similar, or PIs doing similar kind of work for potential collaborations. It can help you find a lot of great information. There's lots of information available on the NIH websites, and I encourage you to explore. Next slide. Neuroscience spans many institutes, and that's why I'm telling you that we have all these institutes. It does, in fact, span a large number of institutes. There are some major players, however. NINDS, the National Institute of Aging, National Institute of Mental Health, National Eye Institute, and National Institute on Drug Abuse are all big players in the space, are all funding grants within the translational space that I work in on a regular basis. And you can see overall the aggregate of all these different neuroscience funding is higher than other disease areas within NIH areas, so higher than cancer, higher than infectious diseases even, when you combine all the different institutes working on neuroscience space and their neuroscience projects. Next slide. So we invest in research across the spectrum. So NIH and NINDS is interested in basic research, and without fundamental neuroscience research and disease-focused research in the basic space, we will never get to translational space and trying to get therapeutics developed. Without clinical, taking it further, many times this will never reach the market so that it can help patients. So we need the whole spectrum. And it is all built on a strong foundation of basic fundamental neuroscience research, fundamental disease-focused research that provides the ideas and the great leaps forward that allow for translation to happen. So we have different divisions in NINDS. We have the Division of Neuroscience. Division of Neuroscience, I'd like to say these are the program directors that have the disease portfolio. And these are the people you want to understand if you want to talk to and understand how your project fits into the greater portfolio in the disease indication area you're talking about. They'll be able to tell you. We have the Division of Translational Research. We really focus on taking that basic research finding and moving it to the clinic. And then they had the Division of Clinical Research is really about moving to the clinic all the way through to FDA review. There's also the Division of Extramural Activities that covers all the spectrum as well, and includes this open group and diversity and training programs. Next slide. NINDS also, has a suite of programs in these different areas. And the ones highlighted in orange here, the ones in translational that we'll probably touch on a little bit through the course of today. We have projects that are in the Ignite space in the very left-hand side, so the preclinical, getting you the preliminary data you need in order to be successful in the therapeutic development programs like the Pain Therapeutic Development Program, like the Blueprint Neurotherapeutics Network. So all of those things I think are wonderful tools. But I'm talking about small molecules today. We have complementary programs for biologics, and there's a typo on the slide. It should be BPN Biologics, not Create Biologics on there. So BPN Biologics is really the only program that's accepting applications presently, and Urgent is for ultra rare diseases and gene therapy. We have device programs. We have programs against countermeasures for chemical threats. We have the large SBIR program that covers a whole gambit of space all the way from preclinical through the clinic for those small business interests out there. And we also have biomarker validation discovery, as well as some services in the screening area, like the preclinical screening platform for pain, as well as the epilepsy therapy screening program. And those fundamentally what the translational space that I'm working in and we're talking about today cover. And they get you through from the basic research finding to the clinic. Next slide. Likewise here, you'll hear translation, and when we're talking today, we're really talking that bench to bedside translation space. The translation space from bedside to the community is in the Division of Clinical Research, and it's the T2, T3, T4 kind of space that you may hear about. As I said before, it all starts with you. You've got to bring us new targets, new small molecules with pharmacology, new ideas for biologics or devices, and we're probably able to help you here and move things forward. Next slide. We help by, you need to establish a collaborative team, and you need experts in the disease biology area, but you also need experts in each aspect of drug discovery and development. You need clinicians that work with the target patient population to inform you what you need to have for a drug in order to be successful. You need to have licensing and business development advisors to help you when to patent, how to get additional investment from outside partners. You can consider partnership through biotech, and there are things like the STTR program or even our SBIR programs that can collaborate with different academic institutions. NIH can help you bridge that gap, and I recommend highly that, again, you reach out to us and talk to us about what those opportunities are. Next slide. We have a number of initiatives, and this highlights a number of them on the slide here, which is like Brain Initiative, the Blueprint for Neuroscience Initiative, the HEAL, Helping in Addiction Long-Term, amongst others. And these programs have money that is many times allocated to those particular disease areas that they're focused on. The Blueprint really covers all of the neuroscience space. Next slide. I'm going to now switch gears and tell you about sort of the Blueprint Neurotherapeutics Network, which is the program I've been working on the longest for the last 11 years. And it started as a grand challenge and as part of the Blueprint for Neuroscience Research. And its goal was to combine the strengths of NIH and industry expertise to small molecule drug discovery, with the goals of de-risking the therapeutics to the point where industry will invest in them, allowing for the potential drugs to reach patients efficiently. We want to identify the best ideas for translation in the community and fund them and also help them with the infrastructure. As I said, you need to have all the pieces to be successful. And to be successful, then the best way to do that is allow us to provide you those resources. So the overarching goal in all these cases is to provide non-dilutive funding, the necessary resources, and to protect your intellectual property so that when you move forward to licensing, it goes in unencumbered fashion. Next slide. The program is for academics and small businesses. The grants are for small molecule discovery, although we do have a parallel track for biologics now. The projects can enter at the discovery phase, meaning I'm still doing optimization of whatever compound or drug entity that I'm starting with. Or I have a development stage projects can come in and say, well, the optimization of the molecule is done. I just need to finish out the development activities on that. There's opportunity to access NIH-funded consultants. These consultants have industry expertise with decades of experience in their subject matter areas. You have access to our contract research organizations that specialize in a number of different areas important to the drug discovery process. And I'll talk a bit more about that in a minute. And you have access to expert NIH project managers with industry and scientific experience, as well as the science officers, those program directors with the disease biology expertise and the understanding of the overall portfolio. And together, we create teams that really move projects forward. Next slide. The BPN has 9 institutes and centers that are signed on to it. And so if you're in the mission of any of these institutions for your particular project, then you could join the BPN by applying through the grant mechanism. Next. As I mentioned, I'll get to this. Every team in the Blueprint Neurotherapeutics Network establishes what we call an LDT, a lead development team. That it consists of the principal investigators and their team, as well as these industry consultants that NIH is paying for that we assign. Those consultants will assign their intellectual property back to the institution of record on the grant. And you have NIH staff there as facilitators. Your organization and your team is responsible for, at a minimum, all the disease biology, the in vitro assays driving SAR, the in vivo animal models of disease. That's got to be as part of the grant budget. If you have access to medicinal chemistry, access to PK, access to toxicology, access to drug manufacturing formulation, access to clinical trial information, or even regulatory stuff, you can utilize those people that you've been working with as part of the grant budget. Most people that come to our project don't have that because they're not in pharma and they're small biotechs that just haven't established that yet. And so they utilize our contracts, and these contracts are industry standard contracts. They're not part of your NIH budget, grant budget. We do that, we pay that directly to them from the NIH side. That allows you to actually stay below some of the limits for grant writing as well. Next slide. As I mentioned, we enter anywhere in the discovery and development pathway, but you can also exit anywhere on that discovery and development pathway too. And we have had projects that have been licensed at the lead optimization stage, at the development stage, at the clinical stage. And so in all cases, we're trying to make sure that whatever we do, if we continue to make the milestones, the project continues to move forward to the clinic. And we've had some grants, some projects where the inflection point is even further than where we go in phase 1, so they continue on grants to go into phase 2. Next slide. These are 2-phase grant mechanisms. We're called cooperative agreements. There's a UG3 or U44 phase 1, followed by a UH3 or U44 phase 2 if it's SBIR. I like to say the preparatory phase is sort of the kicking the tire phase of the program. We really have a go, no go milestone for the first year of the program, showing that, what was in the grant, we can recapitulate, we can get the synthesis up and going, we can do the benchmarking required, we can really determine whether we have a path forward or not in that first year. And then the remaining years work towards the standard set of milestones as we progress the projects forward. Next slide. This is sort of redundant with a lot of what I say. Can you hit one more? Click on the slide deck. Carolyn, just click one more time. Advance one slide, or just advance it a little bit. It's the animation. Thank you. So not all ICs develop projects, will take development projects. So you need to be careful when you come in and say, you know, although I said you can bring them in anywhere in discovery and development pathway, that's true overall for the program, but it's not true for every IC. For instance, NIMH and NIDA do not take development projects under Blueprint Neurotherapeutics Network. The best thing you can do to me, for me, is to call ahead, talk to me about the program, make sure we understand the issues. There are budget restrictions associated with the UG3, UH3, and the U44 that differ. If you're coming in under the UG3, UH3, just make sure that you are covering, if you're over 500,000, you have to ask permission. So covering yourself, making sure that you meet the dates. The next application date is August 9th. Next slide. The milestone progression, I talk about this, these are all milestone driven programs. So as the projects move forward, they're expected to meet milestones every year as we're doing it. They're aligned with the drug discovery and development pathway. This gives you an idea of sort of the timing as we go through the projects. You know, these are based on having resources not only from your lab, but resources from the NIH side through the CROs and consultants to keep you on track and moving things forward. The maximum duration of any of these grants is 5 years for the combination of the UG3, UH3 phase. Next slide. This question always comes up in these programs about inventorship. Inventorship is always determined by US patent law. NIH staff is not, we're there as facilitators. We're not going to be actively involved in telling you which compounds to make or trying to be inventors on your patents. But our CROs are, and our consultants are, and they have to assign intellectual property either by contractual, in the cases of the synthesis folks, and by IP agreement with the consultants. That's why we ask for everyone to have upfront agreements in place between. So if you have multiple institutions on your side, we're going to want to know you have the plan in place prior to award, so that there's no arguments at the end. Next slide. So I want to just wind up and give you a overview that essentially says that this program is great for either small molecules, and we have a complementary program for biologics. For people who want to bring a project forward to do drug discovery and development, develop neurotherapeutics, we can provide you the contract resources in a tailor-made way. We can provide non-dilutive funding. We protect your intellectual property, and we make sure the projects move forward in a industry standard format, so that, again, it is you have all the data you need to have at the end in order to be attractive for future investors. Thank you for your time, and my contact information is here.
Great. Thank you, Chuck. Moving on to Dr. Pellimonter.
Hello, everyone. It's nice to be here, and I am happy to talk to you about a program that is built on the same infrastructure as what Chuck just described, very same infrastructure as Blueprint Neurotherapeutics. However, this program is part of the HEAL initiative and involves some different institutes. So the HEAL initiative, as has been mentioned already, stands for Helping to End Addiction Long Term. HEAL takes two approaches to ending opioid addiction. One is understanding, managing, and treating pain, which is what our focus is today. And the other half is improving prevention and treatment for opioid misuse and addiction. So next slide, please. So I don't think I have to convince anyone that pain is a significant public health problem, and so is addiction. So on the left-hand side of the slide, talk about the addiction piece of it. 90,000 plus drug overdose deaths in 2020. 30%, that represents a 30% increase over 2019. And 75% of those are associated with opioids. So in terms of pain, again, the statistics are pretty jarring. 50 million adults have chronic pain. 25 million have daily severe pain, and 20 million have high impact chronic pain. So next slide. So the HEAL initiative, of course, has research priorities that cover both addiction and enhancing pain management. And so what I want to focus on, of course, is enhancing pain management today. And some of those priorities under that particular category involve understanding the biological underpinnings of chronic pain, accelerating the discovery and preclinical development of non-addictive pain treatments, and that's what we're going to focus on with the Pain Therapeutics Development Program. And then another priority is advancing new non-addictive pain treatments through the clinical pipeline. And you can see in the little slide where the half circle, where we talk about the different priorities under pain management, you'll see basically it's divided into two places: clinical research and preclinical and translational research in pain management. So there's a very large clinical pipeline of potential opportunities for funding in this particular area. The other strategy is to inform the best practices for effective pain management while minimizing the risk of addiction. Okay, next slide. So HEAL programs for enhancing pain management that are focused on accelerating therapeutics through the pipeline actually cover pretty much the whole spectrum of early all the way through early discovery through late development. So on the early discovery end, there's one particular strategy that's about planning for therapeutic development. There are a couple funding opportunities that support that particular phase. One is just focused on basic science discovery and the validation of the target, and the RFA is NS22034. And the other is planning and preliminary biology and mechanism data collection, represented by a funding opportunity RFA NS21029. And as I spit out these numbers, I just want you to realize that you will be, as was stated, you're going to get a copy of these slides. And in the backup portion of these slides, you'll see more information on some of these programs. The next phase that's covered is initial therapeutic discovery. And their RFA NS22052 supports assay development, screening, initial hits characterization, and the development of biomarkers. Another set of funding opportunities or actual resources that are free also support initial therapeutic discovery. They support translational assays, and NCATS is responsible for those. And they also support preclinical therapeutic profiling, and a program called the PSPP, or preclinical screening program for pain, is responsible for the preclinical therapeutic profiling. Another phase is therapeutic development, and there the RFA that we're going to talk about, the Pain Therapeutics Development Program, that supports activities from hit to lead all the way through phase 1 clinical trials. And then finally, we also have funding that supports clinical trials, in particular phase 2, and that's the EPICNet program. And the 10 participating NIH institutes are NINDS, NIAAA, NIDA, NCCIH, NIAMS, NICHD, NIDCR, NIDDK, NEI, and NHLBI. And then in addition to these funding opportunities for research, there's also a training opportunity, PA 20-222, that's the Notice of Special Interest to encourage eligible NIH HEAL awardees to apply for PA 2022 research supplements to promote diversity and health-related research. This is an administrative supplement, and a clinical trial is not allowed for this particular training opportunity. Okay, next slide. So now to really cement that focus on the Pain Therapeutics Development Program. Again, the RFA number is 21010, in case you're interested in looking it up. Like the Blueprint Neurotherapeutics Program, it covers the gamut of early phases of discovery where hits are characterized and leads are optimized all the way to phase 1 clinical trials. It's a UG3 UH3 grant, has been mentioned for BPN, and it has a maximum of 5 years of funding. So some of these characteristics that are listed here are all exactly the same as Chuck just talked to you about for BPN. And the end goals and milestones for this particular funding opportunity are to identify and fully characterize a lead candidate, to identify a target engagement biomarker if possible, definitely to seek partnerships, especially as people move into their IND enabling studies, to complete IND enabling studies, to file an IND, to complete phase 1 clinical trials, and most importantly, to be ready for phase 2 clinical trials in programs like EPICNet. I would say the one big differentiator between the PTDP program and the Blueprint Neurotherapeutics program is that PTDP, in addition to small molecules, it also is open to biologic therapeutics. So all different types of biologics, ranging from antibodies all the way to cell therapy, along with small molecules. Okay, so next slide. Again, similar to BPN, it really is based on a virtual pharma model that features both grant and contract resources. Just as Chuck described, a lead development team is formed when a PI is granted an award for these types of funding opportunities. They and a lead consultant lead the lead development team, and NIH staff coordinate the efforts and provide advice. Program has pretty much the same features as BPN, with the exception, I would say, of the addition of biologics to the mix. Okay, next slide. So eligible organizations for PTDP are academic institutions, small businesses, foreign institutions and components. And then on the topic of foreign organizations that would be applying, reviewers will assess whether the project presents special opportunities for furthering research programs through the use of unusual talent, resources, populations, or environmental conditions that exist in other countries and either are not readily available in the United States or augment existing resources. And so they're in the application, it is really important to explain why the foreign component is uniquely needed for this particular application. Okay, next slide. And again, similar to BPN, applications can enter in the discovery or the development stage. If they enter in the discovery stage, they need to show rigorous biological rationale for the approach. The premise is very important. They need to show that they have a promising starting material, a small molecule or a biologic as a starting point for optimization. And they also need to show that they have scientifically sound assays to optimize and test the agent. It's very difficult to optimize or select a lead candidate without sound biological assays that can be counted upon for reliability. On the development side, the candidate therapeutic has to be identified to enter in this stage. And there should be a strong data package that links the modulation of the therapeutic target by the candidate therapeutic to disease modification. And then biological activity and ADMET properties of the candidate therapeutic must be appropriate for the intended use. And again, regardless of the entry stage, every application should include a list of activities and a description of a plan for both phases, for both UG3 and UH3 phases. And both phases should be represented in the budget as well. Budget should be annual also. These should have milestones that enable the quantitative measurement of success for key grant objectives. And as I mentioned, separate budgets for each year of the grant. And like BPN, there should be a table of activities that indicates what will be conducted by the PI's team and what is requested from the NIH CROs, because as you remember, both Chuck and I were saying that CROs are up to the use of our CROs are there, they're accessible, but they are up to you, the applicants. You can choose which ones you want, or you could choose not to use any at all. Consultants will be provided for you once a lead development team is formed. But you can also propose having your own consultants, and that could be in your budget as well. And then target product profile table has to be in the application, and there should be, biomarkers should be at least addressed in the application, and there should also be a paragraph or two talking about intellectual property plans and you know, whether or not you have permission to use reagents and things like that. Okay, and then all projects should be laying out the path to the clinic via the grant or the NIH contracts. In other words, the reviewers want to see that you are actually looking, you know, forward. You're looking forward to the idea of at least getting into phase 1 with these ideas in this project. Okay, next slide. So now to the 2 milestones, which have been covered for BPN already, but for the PTDP program, they can be a little bit different because, as we mentioned, PTDP looks not only at small molecules as therapeutics, but also biologics, and some of the milestones might be different for biologics. They are an important component for the administrative decision to transition to the UH3 phase. They're used for measuring success and achieving each of the research plan's key objectives. And so for each milestone, when you're writing your application, you should provide details on the assumptions, the experimental designs, and the data analysis plans that go with that milestone. And quantitative criteria for measuring success should also be part of your write-up on each milestone. Quantitative criteria for success will be used in making go, no-go decisions. And so that's how it should be written up. You should think about, you know, what do you absolutely have to see in year 2, let's say, in order to rationalize moving to year 3. And then you should specify the timeline for the milestone, and there should be at least 1 milestone laid out for each year. And then another important thing to remember is milestones will be further refined after the award with the NIH staff and the new lead development team. Okay, next slide. So I think we went through this fairly well on the BPN discussion, and you can see that one thing that's added to our components for UG3 and UH3 are components having to do with biologics development as well as small molecules. So for instance, in the execution phase, you might be doing, in addition to optimizing a small molecule, you might also be looking at things like biodistribution, gene expression, immunogenicity, cell bank development, and so on. Okay, we can move on to the next slide. Now here I just wanted to go over for the Pain Therapeutics Development Program and the funding opportunity associated with that. The basis for withdrawal are the things that are listed in bullet points here. So if you're screening for initial hit identification and that's all your, you know, if that's what the main focus of the grant is, that is not responsive. Same way with proposing basic research and studies of disease mechanism. Also animal model development, not responsive. You need to have your models developed already. Those are some of the biological assays that need to be in place when you apply. In terms of development of diagnostic devices, we have other funding opportunities in Division of Translational Research for those activities. Clinical studies beyond phase 1 testing are also not responsive for this funding opportunity. We will take you through phase 1, but then, you know, that's the hope that you now have the package you need to move to phase 2 using either another funding opportunity like EpicNet or a partnership with industry, whatever way you can do that. And then another thing important with PTDP is that opioid sparing projects and projects targeting the mu opioid receptor are considered non-responsive. So keep that in mind. Okay, next slide. All right, here are just some little hints of things to do in preparing your applications. First and most important is, even though these funding opportunity announcements are long, please read them carefully. And then do discuss your proposal with NIH. It's important to get an idea from the program director and the Home Institute's program scientific experts, just to get an idea if your disease interests, like if we were talking about BPN, it would be really important to know whether your particular disease interest fell within the mission of NINDS or the institutes participating in BPN. For pain therapeutics, that's not as much of an issue because most of the institutes that are involved in pain management are signed on to the pain therapeutics FOA. It's also important if you're in BPN and you're looking at a small business administration grant, a U44, it's important to also contact that person. And I think the rest are all things that we've mentioned already. So let's move on to the next slide. So preliminary and supporting data, it's really important to address the quality of the data that's presented in prior applications in a detailed manner. Were the studies done in a rigorous manner, utilizing randomization, blinding, and so on. So just make sure that when you're talking about your supporting data that's coming from the literature, be critical as you talk about it. And if you're talking about your own data, same way, be critical and just make sure you're very clear and transparent and very clear in your argument about why the premise is important. In terms of rigor, again, detail the controls being used for each type of experiment. Include details within the experimental design about that, reduction of potential bias, and that includes blinding, randomization, and inclusion/exclusion criteria. And then describe the source of the data on which the sample size estimation is based upon. So, in a power analysis, there are multiple variables that are involved. So just make sure you're clear about that. Some other things to do are, move forward to the next slide, please. Complete your required SAMS registrations at least 6 to 8 weeks in advance of receipt dates. Consider submitting your application early. This is always good. And if you're applying to a UG3, UH3, and this is for BPN only, it's not for PTDP. But if you happen to go above 500,000 in direct costs, you need permission to submit and be reviewed. And the request has to be made 6 to 8 weeks prior to the submission date. And again, this is BPN only, not PTDP. And then also, very importantly, talk with your tech transfer and business development group so that you can plan for funding patents and licensing activities. Okay, next slide. Things not to do, don't plan for this to be the sole funding for your lab because as been discussed, milestones are evaluated every year and they can end abruptly if you don't meet them. Please do not under-resource your budget to avoid limits. It's important to be realistic with your budget. Please don't plan for NIH contracts for disease biology because that is what the grant is supposed to fund you to do. And then also, there's no need to put one-off in vitro ADMET experiments into your budget because our contracts can handle like a lot of different types of those in vitro ADMET studies. So if you want to do ADMET, that's fine, but then do a whole, like a whole program in it, supported by your budget. And then please do not code your application as a clinical trial if your plan is to use the NIH contractors. You can actually say no about whether it's a clinical trial if you are using us. And so again, can't stress enough the importance of putting together a table that outlines who is doing what in your application. Okay, then we can move on to the next. So this is so just a little bit off from the funding opportunities for research. We also have an R25 supported grant that provides a training course in neurotherapeutic discovery and development. It's called Training in Neurotherapeutics Discovery and Development for Academic Scientists. This is in the process of, I think, being renewed right now. It's covers small molecules and biologics. It covers various topics critical to discovery and development. It provides individualized mentoring and assessment, and it provides opportunities to interact with topic experts. It has an online lecture series. Lots of people have participated to date. And representation has been over 50 different institutions and universities, and disease representation also from multiple NIH institutes. So this is a very important course if you're interested in just learning about discovery and development. All right, next slide.
Okay. So I think we can take some questions. Let me check out the chat. So far, we have one question. What's the budget cap for the U19 HEAL proposal?
$1.5 million in direct, including sub consortiums and as well, according to the RFA. It says application budgets are limited to direct costs of $1.5 million per year, including all consortium and sub-award costs.
Okay. Still waiting for questions.
I do want to clarify one thing that Marianne said, just to not have people put it in the budget backwards. The in vitro admit, the one-off experiments, you should include in your budgets. So if you want an in vitro admit program to be supported by BPN contract, we can easily do that. But if you want it to be supported like a single experiment, like I want to do a syrup screen, and I want BPN to just do that one thing, that's something you should just contract yourself, because the amount of time and effort for us to put a contract in place, it just costs too much to do that for that one experiment.
Okay, we'll give it. Okay, you said that the entirety of the clinical trial could be contracted out to NIH CROs without including those costs in the grant budget. Is that the case, and how would that work?
Yeah, that's correct for phase 1 only, not all phases of clinical trial. So it's really primarily the single ascending dose, and we can also support multiple ascending dose, but that's it for phase 1. And it is true, if you want us to do that, then you do not have to put in any details about that in your application. You just have to specify that NIH will do that for you.
Yeah, I mean, that's true also for Blueprint Neurotherapeutics, that we can support the thing. So just as a note, if you're going to be doing the clinical trial through the NIH contract, it's not a clinical trial grant, and it's not a human subject grant, unless you have something else going on to qualify for that. All the paperwork associated with clinical trial paperwork is going to be submitted as part of the contract process. So the Form E's and things that you might be interested or used to for filing a clinical trial, that stuff would be dealt with there. It is important to note that if you're thinking about patient populations, especially small patient populations that are only available in certain areas, again, that's not something the NIH contract can do easily. So you should talk to us about what you want to actually accomplish.
And your resources would help design the study as well?
Yeah, absolutely. Most phase 1 clinical trials start with healthy volunteers, and they're usually doing what is essentially human PK, as Marianne pointed out, in the single ascending dose studies. That's based off of your toxicology and your efficacy. So you're going to look at some, usually a multiple of at least 10 below where your efficacious dose is, and start to work yourself up from there, and no higher than your predicted maximum tolerated dose based on your tox trials.
Okay, for the regulatory strategy and generation of IND package for phase 1, does the NIH assist, or do we need to do all internally and work with our own regulatory consultants?
So the NIH will help. I mean, with the consultants that they can provide, we do have regulatory affairs consultants that we can assign to your lead development team. But it is also absolutely fine to include your own. Usually everybody works together as part of the lead development team on that.
Yeah, it's important to note that the consultants that are working with you throughout this process will help write the certain sections of the IND package that are relevant to their expertise. So the toxicology consultant will help with assembling the toxicology section of the IND. The DMPK people will help in the non-clinical overview section. But at the end of the day, you definitely need to have something in your grant budget for regulatory filing. Because we're not going to file the IND. Your institution's going to file the IND. So there needs to be some money in the budget, no matter whether we're funding all of the regulatory IND preparation. The final QC and filing is going to be your responsibility, your institution's responsibility, because you will be the sponsor of record.
As a trainee, how do I prepare or work towards participating in some of the programs mentioned?
So I would suggest the first step is to look for the training courses like the one that I mentioned, or the diversity training course. I mean, I think it's important just to take those courses, and then as a trainee, actually maybe try to find mentors who you can work with who are actively participating in drug discovery. You'll hear something about programs like that from our awardees like Jerry Rook and Takashi Tsukamoto.
Okay. Can you explain why screening to identify hit compounds is non-responsive?
Right. So remember I said it's non-responsive if that is the focus of your grant. We have other funding opportunities that specifically support those types of activities. One of the things we expect for our entry criteria is that your screening assays are developed and validated, both in vitro and in vivo. But sometimes we realize that sometimes you have to optimize assays while you're actually developing the therapeutic, and that's fine. But all I'm saying is if that's the primary focus, then it's not responsive.
Yeah, I mean, if you're within NINDS, as mentioned, for instance, there's the program called Ignite. Ignite has a funding opportunity, I'll paste it in, PAR 21-124, that is designed around developing an assay and running a screen to identify a therapeutic agent. That allows you to do that screen. You're talking about develop an assay, run a high throughput screen, for instance, and find your starting point. Additionally, the Ignite program has another FOA that is related to it that allows you to do early stage optimization of that hit and develop in vivo biology data associated with that. Gets you the preliminary data to make a more compelling application for BPN or the PTTP program.
And then I want to point out to Dr. Julia Bachman just put into the chat also on the HEAL side, on the pain therapeutic side, there's also a funding opportunity that's a U19 grant mechanism that supports that exact thing as well, supports assay development. That one is 22052, and you can find it in the chat.
I think that's it. I think, we have, yes, I will share a link. We will share the slides and the links to anything we mentioned afterwards. So Marianne, let's move on to the next part of the talk, shall we?
Sure, that seems like a good idea. All right, so Everyone, I think one of the exciting parts of this webinar, in addition, of course, to listening to information about our funding opportunities, is a chance to hear from people who have been awarded our funding opportunities and their experiences. So our panelist list of awardees include Dr. Jerry Rook, who is the Associate Professor of Pharmacology at Vanderbilt Center for Neuroscience Drug Discovery, Vanderbilt University, and then Dr. Takashi Tsukamoto, who's the Associate Professor of Neurology, Brain Science Institute at the Johns Hopkins University. So I think they'll both be giving you some very interesting perspectives on drug discovery in an academic setting. All right, Dr. Rook. Yeah.
Thank you. First of all, I'd like to thank Chuck and Marianne for giving me the opportunity to share with you my career development and my experiences as a HEALS initiative awardee through the NINDS. I've been actively involved with drug discovery research for the past 14 years. So just to give you a brief overview of my career development, I received my PhD in the Department of Pharmacology, Toxicology, and Therapeutics from the University of Kansas Medical Center in 2008. My thesis work centered around the crosstalk between pain, neurocircuitry, and the immune system. So it prepared me well for this award. I then wanted to pursue a postdoc specifically in training for drug discovery. So with the guidance of one of my advisors, Dr. Sam Enna, whom many of you may know, he advised me to come join the Center for Neuroscience Drug Discovery at Vanderbilt University in the lab of Jeff Kahn, who was the founding director of the center. And so Jeff came to Vanderbilt from Merck, had extensive experience in drug discovery, and he started the center back in 2003, I believe. So I joined his lab in 2008. And while my postdoc was a typical academic postdoc, what was unique about it is that all of our trainees, our projects are centered around this initial target discovery or target validation for our drug discovery programs. So at a very early stage in my postdoc career, I began being exposed to drug discovery programs. And like most people who want to get in drug discovery, I had always believed that I would eventually end up in industry because at the time, there really wasn't a great amount of academic drug discovery programs out there. So during my postdoc, I was fortunate enough to get a spot in the training program that Marianne had discussed, the Neurotherapeutics in Discovery and Development. And if you have the opportunity, I highly recommend that you apply for this program. It's an extremely well laid out, inclusive program in drug discovery. It goes from the very beginning of target validation all the way through getting a drug to the market. And it's taught by experts in the field of drug discovery, both from an academic and from an industry setting. So you get both perspectives on that. And I was able to participate that in my postdoc. And then in 2014, I decided to go ahead and stay in academia and join the pharmacology department at Vanderbilt and the Center for Neuroscience Drug Discovery, where I was the director of the behavioral and in vivo pharmacology. And so that's the background of my training. I'm now associate professor in the Department of Pharmacology. And in 2019, again, fortunate enough to be able to receive a HEALS Initiative Award from the NINDS. And this is to help develop a novel pain therapeutic that's non-addictive. So our target is focused on the metabotropic glutamate receptor subtype 5 negative allosteric modulators for the treatment of chronic pain. And at the time of our award, we had already had a significant amount of target validation data to support the pursuit of this target. Both preclinical and clinical data have demonstrated an analgesic effect of these compounds. We had already validated our high throughput screening assay, had ran a high throughput screening, and had a series of chemical matter around this target. So in 2019, when we received the U grant, we were really in the lead optimization stage of the program. And so with the help of NINDS, we were actually able to successfully navigate lead optimization. So during the first 2 years, we generated 11 new chemical series, which produced 8 new patents around the target. We were able to declare a clinical candidate. So we now have a clinical candidate. We've transitioned from the UG3 to the UH3 portion of the award. And so now we're in the process with the support of NINDS to complete our pharmacokinetic, our in vivo pharmacokinetic studies, some efficacy assays, as well as, of course, addiction assays to demonstrate non-addictive properties of this compound. And we're fortunate enough, Marianne had mentioned this, that it's extremely important to have targeted engagement. And we're fortunate enough for this target to have a PET radioligand. And so we're in the process of doing receptor occupancy studies in rodents to establish our efficacy and receptor occupancy relationship, and that we will use those data to then generate our human dose projections as we project through into the clinic. And during the UH3, we also opted to go through the NIH CROs. So all the safety pharmacology and toxicology studies that Chuck and Marianne have mentioned are these pre-IND enabling studies are all budgeted through the NIH. So those weren't included in our budget award period. The UG3 phase, for those, we budgeted for all of that internally, either through the Warren Center for Neuroscience Drug Discovery at Vanderbilt or with our collaborator, Rob Giroux, who's the director of the Pain Center at Washington University in St. Louis. And so now we're just working through our safety pharmacology toxicology studies and hopefully progressing to submit an IND application within the next 2 years. And with that, I guess I'll stop and take any questions anyone has.
Yeah, we're going to move to the next speaker, and then we're going to go to questions. So go ahead, Takashi.
Hi, so my name is Takashi Tsukamoto. I'm a principal investigator of UG3 project titled Development of MRGPRX1 Positive Allosteric Modulators. As Marianne and Chuck mentioned, so this is one of the really important mechanisms that which is really sort of played as a sweet spot for our project. But before I get to that, I put all the elements that they wanted us me to put. So I'm just to go through this. Oh, I didn't know that this one has an animation. So, I'm trained as a synthetic organic chemist at Tokyo Institute of Technology. This is where I got the both bachelor and PhD degree. And then I have been fortunate enough to get the position as a postdoctoral fellow at University of Michigan. Maybe you can click, that will show you the Michigan position. Maybe not, I guess. So that's okay. And so this is where I got the postdoctoral position at College of Pharmacy. And that's where I was really exposed to the interdisciplinary environment, because University of Michigan, School of Medicine, Chemistry, Pharmacy, they're all in a one campus walking distance. So I was able to participate in lots of different seminars, collaborators, and that really inspired me to get involved in the drug discovery field. So after I finished my postdoc, I got my first real job as a medicinal chemist at a company called Guilford Pharmaceutical. That's in Baltimore. And the company was acquired twice, once by the company called MRJ Pharma, and the second time by the Japanese company called Eisai Pharmaceutical. So I didn't really change the job, but I had an experience with 3 different companies. They all have a different culture, different size, different strategies. So it was a great learning experience for me as an industrial chemist. So in total, I spent 12 years in the industry section, and then I moved to Johns Hopkins University. So one of the reasons is Eisai chose to close the site in Baltimore. So I had option to move to Boston, but around the same time, Johns Hopkins School of Medicine was actually interested in creating our own drug discovery unit within the medical campus. So I thought, oh, I didn't really have to at least move. So some of us together from ASI moved to the Johns Hopkins as a member of Johns Hopkins drug discovery team. So the mission of this group is really work collaboratively with the Johns Hopkins faculty members who may have really exciting basic science that has a potential to translate into a drug discovery project. And this particular project, MRGPRX1, is really one of them. So now I'm going to move on to the how our project went from the idea to funded project. So at Johns Hopkins, I met with Dr. Jin-Jun Dong. He's a professor at neuroscience. And he had this interesting G-protein couple receptor called MRGPRX1. This is a receptor that is specifically expressed in DRG sensory neuron, which makes this ideal target for pain. So, and then we initiated cell-based high-throughput screening a couple years later. And in 2014, we actually are fortunate to identify both MRGPRX1 agonists and allosteric modulators, so-called PAMs. And in 2016, prior to getting this HUE grant, we received this R21 grant to conduct a lead optimization for the hit compounds that we obtained from the screening. And that leads to the quite interesting lead compound that we think we can further improve to the therapeutic agents. And this is when we became aware of this interesting opportunity based on the UG3, UH3. As Chuck mentioned, the first thing you need to do is call NIH to try to figure out the fitness of this project. That's exactly what we did. So early 2019, we had the teleconference with Chuck, and that really convinced us this is the perfect mechanism for us to move this project forward. So we've been funded since the end of 2019. So we are currently in a third year, which means the first year of the UH3 phase for this project. So research and support critical to our success. I think most important thing is probably this one is obvious. You just have to have great people to work with. So I was really fortunate to have very productive lab members, Niara Hing and Ilyas Burhan Ajit. And of course, the collaborator, Jin-Jun Dong, is the one that brought this target. Yung Guan is the expert in animal pain models. And Dr. Raja is the very seasoned pain clinicians. Also for this project, since this one is based on cooperation with the NIH team, we had the NIH team that assembled by NIH end. And Becky Roof is the one that we are supporting our project from the beginning. And we also were fortunate, like Vanderbilt, we have a very drug discovery oriented team where we have some resources available to do some core studies related to the drug discovery, including simple metabolism studies and pharmacokinetics and so on. And this one is led by the head of the Johns Hopkins Drug Discovery, Dr. Barbara Strasser. So I was really fortunate to be surrounded by those people, and especially that what makes this one much easier for me was that I was able to identify all these people at Johns Hopkins. So my experiences, for the last 2.5 years as awardee of this UG3 and UH3 grant. And one thing that makes this very different from other grants that I received in the past, like R21, R01, is that this is a really milestone driven. So you have this predetermined milestone that given at every stage, and you really have to go through this. So you can't kind of take a detour or do something because you think it's interesting. That's not how this project is operated. So you got to really always look at the milestones. And we have actually meeting now every 2 weeks to really go about how we are making progress towards those milestones. Like any other drug discovery project, we had lots of ups and downs in our project. And it's also important to be very transparent about the problems you have with the NIH stuff. And one of the reasons is they are here to help us. So they are not here to review our project. So whenever we bring up those projects and issues, they are on our side, we provide potential solutions or how we can change the direction of the research. So it's very important to be transparent. And that also leads to the another important thing. This is a absolutely team effort. So collaborative, and we are actually working almost as like one team, and that makes this project even more productive. And it's really exciting to have all these people, especially those people that provide expertise outside of my expertise as a medicinal chemist. And this one is also a great learning experience for me personally, because as a medicinal chemist, I typically involved in a very clinical stage of the drug discovery, but now we are getting into the area where we're picking compound, doing further animal studies. And those are not something I had a great experience when I was an industry chemist. So I'm learning every 2 weeks when I meet with the NIH team at our conference call. It's also a great training experience for our chemists, particularly Dr. Ilyas Parhan, that's showing us second photos. He joined as a postdoc to our group just after we started this project. And he actually had opportunity to participate in those calls we have with NIH. And this was one of the best experience he told me that he had, because he was purely trained as an organic chemist during his PhD program. And this really inspired him to open up his skills into a different areas of healthcare field. And as a matter of fact, I think it's a month ago, he found a full-time job as a consultant in healthcare medicine. And he really appreciated the experience he had and all the things he learned through this project. So, so there's lots of impact besides the progressional project that this HEAL project offered us. So I'm really fortunate to be a part of this and really appreciate the support by NINDS, especially also the support given by some of the staff, including Chuck and Miriam, and most importantly, Becky Roof, who is always with us. And we have a constant meeting besides every 2-week meeting to really try to figure out the best way to move forward our project. I think that's all I have. Thank you.
Thank you so much. That was great. Thank you to both of our panelists. We would be happy to have a discussion now. If anyone has questions, please put them in the chat. I'm going to check the chat. We don't have anything right now. Do each of you want to answer what is something unexpected that you have learned from the program?
Let me think. Something unexpected. I think, I don't know if it's unexpected. I was pleasantly, I don't even know if I'd say surprised, but the amount of expertise that's available through the NINDS and the HEALS initiative that I'm involved in, it's extremely impressive. So the one great thing about having the lead development team is that regardless of what stage you're at in the drug discovery and development process, you can reach out to your program officer or your development team, and they will find the resources that you require to progress the program. If it's questions, if it's studies that need to be conducted to progress the program, there's options for adding additional studies if needed, and different funding mechanisms through supplements that can help progress your project forward, which a lot of times you don't get that kind of support through other funding mechanisms. So I think that would be the one surprise, I guess, that we had when we joined this initiative.
Thank you.
So for me, when I was awarded this grant, I was really envisioning this is like another R01 grant with more money. So I was just really trying to, okay, let's start working on this project. And as I do for other projects, but I wasn't aware of this strictly milestone-based mechanism that they have. And I was given the list of those milestones at our first meeting. And I was like, wow, this is very different. And I was a little overwhelmed in the beginning, but actually those milestones really help you to stick with your plan. And I think it really made us more productive and focused and really object-driven. And this also helps to my collaborators because they are in a basic science field. So they are not really accustomed to those drug discovery type driven mechanism, but because the milestone that we have, that they were able to align with our objective and really move this project forward. So that's one of the biggest difference between this grant versus other research-driven grants that I have received in the past.
Those are great answers. For both speakers, having gone through the program, is there something you wish you would have known before applying or when writing your application?
So for me, I believe it was either Chuck or Marianne addressed this. I wish I would have put a little bit more thought, I guess, into the budget for the first phase because, you know, our lead optimization took about a year longer than we anticipated it would. And so we had, you know, we had under budgeted for the first phase because it took longer than we initially thought it would take. So take their advice, you know, to heart when they say really carefully sit down and think about your budget and anticipate having, you know, maybe additional studies that you'll have to, or questions that you'll have to address. So that is probably the only thing that I would say that I really wish I would have thought about a little bit before.
And for me, I think I can echo what you said, but yeah, those are also budget. It's difficult to kind of put the precise 5-year budget for the drug discovery because it could fail anytime or you need to have a change in the direction all the time. So budget was one challenging thing. I think the other thing I wish I knew better is the variety of pharmacokinetics in vitro ADME studies that's available. So I was very naive there and just have a in vitro metabolic stability. That's all I need. But I'm learning a lot through NIH consultants that there's so many different kinds of ADME characterization available. And they're not just available, they're very useful in really predicting what likely to happen in a human. So I thought probably I could have articulated my grants better if I knew those new technologies in the ADME field. Yeah, but it's okay because this NIH consultant we have, his name is Ron Franklin, he's very helpful. And we are actually making a good job in running all these available ADME studies. Mm-hmm.
What would you say was your biggest hurdle in your path to drug discovery?
Regarding career development or this specific program? So for my career, I would say that, and this has changed over the course of my career, but early in my career when I was trying to do drug discovery in academia, it was extremely difficult to try to find funding mechanisms to support the research. That's changed astronomically, honestly, due largely to the support of NIH now for the support for academic drug discovery. So there are several mechanisms now that can support our research that's focused on drug discovery and not just hypothesis, driven science to get us translated from, Chuck said, the bench to the bedside.
Can you repeat the question?
Is there any challenge or hurdle that you've had in your career when pursuing drug discovery that you've had to overcome?
Yeah, I think the majority of my career as a medicinal chemist, I've been in the neuroscience space. And as you know, that the drug discovery and development in the neuroscience space is pretty difficult. I think one of the needs is probably there's so much unknown in how brain function. So, and that makes it more difficult. So I think that's one of the challenges that I have encountered since I started my career. And even now, I think that's probably the same. And also, I think one of the challenges we have now is really whether still there's an appetite on the industry side to develop pain therapeutics, because there's so many failures, and I'm just concerned they are not really as excited as they used to be about the new opportunity or new ideas that targeting pain.
Thank you both. Sorry if you mentioned this before, Dr. Rook, but how much did your 5 years of planned activities deviate from your original application? And was there adaptability within NIH for changes that came up?
So I would say that for the project, the outline studies in the original application did not change. It was the timeline that had the largest change, in that we really anticipated that we would be able to declare a candidate within the first year, and that unfortunately took an additional year. Because we were still meeting milestones, the program continued and progressed, and we were able to get to a clinical candidate by the end of the second year. So I would say the NIH has been completely supportive and adaptable throughout the process, even with the delayed timeline, as long as we were continuing to make that, that progress. We have had some things happen. I would say one of the biggest things is that, and which was supported by the NIH, that we didn't budget for or outline in our application, was we ran into some solubility issues. And so the solubility with our free base of our compound as small molecules is not the best. So now we have initiated a polymorph salt screen for the program to try to help choose a salt form that's able to increase our solubility. And that's completely supported by the CROs through NINDS.
Chuck and Marianne, do you want to add anything to that?
I don't have anything more to add to that particular, that question or anything, but I was just curious between the two of you, is there any area of expertise that you feel you wish you had from the NIH, but you do not have?
No, I can't say that I've ran into that experience yet, Marianne. You all have been fantastic at finding the resources that we require, so. Okay.
Takashi, what about you? Just curious.
So I believe this particular mechanism, we are the one that has to take care of the manufacturing of the drug for especially non-GLP and GLP studies. Now, we budgeted, but I'm not the expert in this area, so I wasn't entirely sure if I budget that correctly. And also, I'm not really qualified to overseeing all these manufacturing operations. So that's one of the concerns I have as we move forward in this project. And of course, it will be nice that the manufacturing part is also handled by the NIH side.
Okay, I think we have 5 minutes left, but we have one more question. Can you describe the initial outreach call to the program director? Was it just you and an abstract? Was it a team with a presentation? How were you received?
Who's that directed to, Carolyn?
Can you?
Go? Oh,
yeah.
You want to go ahead?
You can go first.
So I got this email from NINDS about the webinar about this UG3, UH3 mechanism. I think Chuck is the one that presented. And right after the meeting, I asked for the teleconference with him. I think that was before Zoom was available, because right before COVID. So I think we just had a teleconference. But yes, we put together the slide deck to go over our project, where we are, and a few questions. And we had a teleconference. And right after that, I think we felt that that this is a perfect opportunity for us. Chuck was really kind enough to go over some of the key points that needs to be in proposals and how it can be written. So he was very helpful. So I have been contacted in the past by a lot of other investigators, both Hopkins and outside, because about, how did you get this Hill grant? What can I do? So always I just say, just call Chuck. That's what I say.
That's what I say too. You want to say anything, Dr. Rick, about reaching out or how you connected with the program?
Yeah, so for me, I drafted a set of aims and emailed Chuck. And so that initiated the discussions. And again, he helped, focus, aims, outline the different budgets that were available as far as whether you budget internally or through CROs, et cetera. And so that's what started the initial application.
Great. Okay, well, I want to thank Dr. Saiwen, Dr. Pelley-Mounter, Dr. Rook, and Dr. Tsukumoto for a wonderful webinar. And thank you all for your questions, and have a wonderful day.
Thank you for having us. Bye-bye.
Thank you all so much. I will just add into the chat, our next webinar is on September 1st on neural engineering research opportunities with NINDS engineering program staff. And we look forward to seeing you next time. And I will send an email with the recording and all of these resources that we shared today. So thank you.